Hello, UCLA. Welcome to our presentation. My name is Pranil,
I'm Ritu.
I'm Caroline.
I'm Naya. And I'm Sunny.
and we are here with Jacob's Lab to present the work that we've been doing for the past year on pre-disease dysbiosis as a driver of intestinal inflammation in patients with inflammatory bowel disease. First, Caroline is going to go over some background and context for our project.
Yeah, so what is IBD? IBD is a chronic inflammation of the gut that doesn't have a known infectious trigger, which means it doesn't have a specific pathogen that causes it. There are actually two types of IBD. The first is Crohn's disease, which is characterized by transmural inflammation of usually the small intestine. So transmural here just means that all layers of the tissue are inflamed. The second type is ulcerative colitis, which is characterized by mucosa inflammation of the colon and rectum. So in this type, only the top layer, which is the mucosa, is inflamed. So as our diets change and lifestyles modernize, the incidence of IBD is expected to go up, which is why we're kind of doing this study. Specifically, our study is looking at the relationship between dysbiosis, which is an altered microbiome composition, and IBD. Specifically in IBD, the ratio of firmicutes and Bacteroidetes, which are two species of bacteria, is altered, which creates an environment that's really conducive to inflammation. So in our because previous research has shown that there is a strong correlation between dysbiosis and IBD, but the exact sequence is unknown. We're trying to determine whether dysbiosis causes IBD or if IBD actually causes dysbiosis.
So like Caroline mentioned earlier, the objective of our research project is to causally link pre-disease dysbiosis to increased susceptibility for intestinal inflammation in patients specifically diagnosed with ulcerative colitis. So in order to carry out this project, this is the outline that we used for our experiments. So first, we had to identify control and experimental microbial community states, otherwise known as OTUs. In order to establish a causal relationship, the experimental OTU type must indicate dysbiosis regardless of IBD status. Then we were able to generate IL-10 knockout mice, IL-10 being a core cytokine responsible for controlling inflammation during the immune response, and we inoculated the mice with these previously identified human OTU types using a fecal transplant. Once the mice were allowed to develop in an entirely germ-free environment, we were able to isolate the mouse tissues, specifically the duodenum, jejunum, and ileum of the small intestine and the colon of the large intestine, and we separated the tissue with and without stool. In our specific project, we stained the tissue with stool, and the tissue without stool was sent to analysis for white blood cells that might reside within the tissue. Then we were able to image and analyze the results. We are currently in the process of imaging some of our results and conducting some of the analysis using IMAGE-J. So first, to determine the OTU type associated with dysbiosis for our project, we had to identify first-degree relatives of IBD patients because these relatives had OTUs, otherwise known as microbial community compositions, that reflected a dysbiotic state, but were not necessarily diagnosed with IBD. And so the first group in our experiment acts as the control, OTU type 1 as a wild-type microbial state, and our second OTU type acted as the experimental state, which reflects dysbiosis, specifically from a cohort of 21 families with histories of ulcerative colitis. Using this humanized gnotobiotic mouse model, we were able to isolate these specific OTU types from fecal samples taken from both unaffected individuals and individuals diagnosed with ulcerative colitis. And then we were able to inoculate all 4 different categories of our IL-10 knockout mice. After allowing them to develop for about 12 weeks and staining the tissues using a hematoxylin and picoseries red procedure, we used a microscope to image the slides. And like I previously mentioned, we are currently undergoing analysis. In regards to the iron hematoxylin and picoseries red stain procedure, this was extremely helpful for our specific project because picoseries red specifically binds to collagen, which is indicative of fibrosis. Using the picoseries red stain allowed us to quantify the amount of fibrosis in the tissue and better establish a causal relationship between dysbiosis and greater inflammation in patients diagnosed with ulcerative colitis.
Hello. So I will be talking about the results that we presented. So first and foremost, I'll go through a quick diagram of the anatomy. So first, on the very top of the slide, we have the stomach, which is not pictured here. Then we have the three parts of the small intestine, the duodenum, which is the first part, the jejunum, which you see the second part, and the third part, which is the ileum, and then that leads to the large intestine. So the slides that we measured and looked at were the parts of the small intestine as well as the colon. So we can see from this first slide the different parts of the slide makeup. So first we have the basement membrane, which is the very bottom of the slide. It's a yellow sort of layer, which is made up of proteoglycans, calcium proteins, and collagen proteins. And then we have that red layer above that, which shows the marker of collagen, which is a kind of indication of inflammation and fibrotic tissue, which we can see is stained on the left of the slide of the duodenum. Then we have the submucosa, which is kind of underlying the mucosal layer. And then we have the mucosa, which is near the intestinal villi. And the mucosa is the kind of area that's most damaged in things like ulcerative colitis. And that's the area that's most inflamed. So next slide, we have the jejunum, which was stained similarly. We can see the red stains indicate the fibrotic tissue and the inflammation. Another slide of the jejunum. Then we have the ileum, which you can also see red fibrotic tissue in both slides. And then in the colon, we can see the sort of villi look slightly different here. We have the mucosa, which make up the villi, submucosa, and then basement membrane on the very bottom. So thank you guys. Thanks for listening.
In terms of the analysis, we're kind of focusing on the fibrosis staining, which is indicating the amount of accumulation of scar tissues on the intestinal walls and on the different parts of the intestine that we have already isolated. And when we're doing the staining, the analysis of the staining, the slides are all mixed up between the OTU type 1 and OTU type 2, so we aren't able to determine just by looking on the slide which OTU they're part of. And we're still currently working through the scoring and analysis of all of the slides, so we won't be able to determine how significant each of the values that we obtain are until we finish everything. And when we put an image through image J, we get 3 of the more important numbers, so we kind of get the area, standard deviation, but most importantly, we're looking at the percent area of the stain, which is kind of telling us how much fibrosis we have because it measures the percent of the red area compared to the area in total. So this is an example for the duodenum. You can kind of see that the percent area here will be 7.44%, which is kind of just the amount of redness you see compared to the entirety of the part that we outlined. So when we're looking at this, we're not looking at the entirety of the slide, just specifically the parts that were traced out. And this is another example for the jejunum. So you can kind of see that the white lines kind of indicate the total area. And then here we get a higher percent area at about 12.231%, indicating more fibrosis. And then the ileum, we kind of get an 8.44%, which is slightly lower than the last slide. And then for our final slide, we kind of get a percent area for 9.465% for the colon. So we can kind of see that just based off of these different slides for each of the section, the highest amount of fibrosis we got was with the jejunum.
So thus far, most outside research papers have indicated there's just an association between dysbiosis and inflammation, but the main question still remains on whether this is merely a correlation or it can be considered a causal relationship. And as mentioned in the previous few slides, we're still continuing to image and analyze the isolated tissues from the inoculated mice in the study. Afterwards, we'll be able to compare this fibrosis observed in these results to the wild-type mice, and we hope to eventually find evidence that knockout of IL-10 does lead to intestinal inflammation in IBD. Outside of completing the analysis directly related to this study, future directions could also look at other forms of inducing dysbiosis. For example, overexpression of TL1A is one form, and we're currently actually conducting a study, again, using hematoxylin and picrocereus red stained tissue to address this. Then we'll be able to determine if other mechanisms of dysbiosis can lead to inflammation.
That is it for our presentation. Thank you so much for your time and listening. If you have any questions, you can field them to my email below or field them to the Jacobs Lab. Thank you.